Fossil insects. And extinct orders. Submitted by Amala Paul. Second year MS. Zoology. Fossil insects. Hollow diamond containing filled diamond. A fossil is any preserved remains, impression, or trace of any once living thing from a past geological age. Hollow diamond containing filled diamond. The insects were trapped, caught in mud or sticky resin, and thereby left a permanent record. Hollow diamond containing filled diamond. Our knowledge of their evolution. About 12,000 species of fossil insects have been described. Countless thousands of specimens have been collected. Hollow diamond containing filled diamond. Fossil insects are not found in as many deposits or localities as most other invertebrates. Hollow diamond containing filled diamond. Like other organisms, insects are preserved as fossils by a sequence of events that results in their burial in a suitable medium. Hollow diamond containing filled diamond. Immediate burial is necessary to preserve the whole insect, otherwise the body parts soften and fall apart, and only the wings remain. Hollow diamond containing filled diamond. The wings decompose more slowly and therefore can be preserved under less favorable conditions. That is the reason why many specimens of fossil insects consist of wings alone. Hollow diamond containing filled diamond. When conditions were good for preserving insects, large numbers of fossils usually occur. Hollow diamond containing filled diamond. Fossil insects have been found at nearly 150 localities in various parts of the world. Hollow diamond containing filled diamond. The oldest fossil insects are primitive, wingless insects similar to modern silverfish and springtails that lived during the early part of the Middle Devonian period, roughly 395 Maya. Hollow diamond containing filled diamond. These primitive insects are known only from a few specimens found in rocks from Scotland, New York, and Quebec. Hollow diamond containing filled diamond. Nonetheless, these fragments have led experts to agree that insects must have originated millions of years earlier, perhaps at the end of the Silurian period, about 419 million years ago. Hollow diamond containing filled diamond. The first substantial record of insects is in Pennsylvanian rocks, deposited from about 323 to 299 Maya, by which time insects had evolved wings and a variety of other specialized structures. Hollow diamond containing filled diamond. Between the scarce Devonian fossils of primitive forms and the abundant and diverse Pennsylvanian fossils, there is a huge gap in the fossil record. Hollow diamond containing filled diamond. So far, no fossil insects are known from Mississippian rocks, most of which were deposited in marine environments. Hollow diamond containing filled diamond. Thus, both the origin of insects and the evolution of wings remains a mystery. Hollow diamond containing filled diamond. Preserved in amber. Much of what we know about prehistoric insects is derived from evidence trapped in amber, or ancient tree resin. Hollow diamond containing filled diamond. Because tree resin is a sticky substance, insects would quickly become trapped upon landing on the weeping resin. As the resin continued to ooze, it would soon encase the insect, preserving its body. Hollow diamond containing filled diamond. Amber inclusions date as far back as the Carboniferous period. Hollow diamond containing filled diamond. The insect evidence recorded in amber documents the relationship between ancient insects and forests. Put simply, insects trapped in amber lived in or near wooded areas. Impressions. Hollow diamond containing filled diamond. An impression fossil is a mold of an ancient insect, or more often, a part of an ancient insect. Hollow diamond containing filled diamond. The most durable parts of the insect, the hard sclerites and wings, comprise the majority of impression fossils. Hollow diamond containing filled diamond. Because impressions are just a mold of an object that was once pressed in the mud, and not the object itself, these fossils assume the color of the minerals in which they are formed. Hollow diamond containing filled diamond. Typically, insect impressions include only a mold of the wing, frequently with sufficiently detailed wing venation to identify the organism to order or even family. Hollow diamond containing filled diamond. Impression fossils date back to the Carboniferous period, providing scientists with snapshots of insect life from up to 299 million years ago. Compressions. Hollow diamond containing filled diamond. Some fossil evidence formed when the insect, or part of the insect, was physically compressed in sedimentary rock. Hollow diamond containing filled diamond. In a compression, the fossil contains organic matter from the insect. These organic residues in the rock retain their color, so the fossilized organism is conspicuous. Hollow diamond containing filled diamond. Chitin, which makes up part of the insect's cuticle, is a very durable substance. When the rest of the insect body decays, the chitinous components often remain. Hollow diamond containing filled diamond. These structures, such as the hard wing covers of beetles, comprise most of the fossil record of insects found as compressions. Hollow diamond containing filled diamond. Like impressions, compression fossils date back as far as the Carboniferous period. Trace fossils. Hollow diamond containing filled diamond. Trace fossils capture clues to how insects lived in different geologic time periods. Hollow diamond containing filled diamond. Here fossilization can preserve burrows, frass, larval cases, and galls. Hollow diamond containing filled diamond. Trace fossils provide some of the richest information about the co-evolution of plants and insects. Hollow diamond containing filled diamond. Leaves and stems with obvious insect-feeding damage comprise some of the most abundant fossil evidence. Hollow diamond containing filled diamond. The trails of leaf miners, too, are captured in stone. Mineral replications. Hollow diamond containing filled diamond. In some fossil beds, paleontologists find perfect mineralized copies of insects. Hollow diamond containing filled diamond. As the insect's body decayed, dissolved minerals precipitated out of solution, filling the void left as the body disintegrated. Hollow diamond containing filled diamond. A mineral replication is an accurate and often detailed three-dimensional replica of the organism, in part or whole. Hollow diamond containing filled diamond. Such fossils typically form in places where water is rich with minerals, so animals represented by mineral replications are often marine species. Hollow diamond containing filled diamond. Mineral replications give paleontologists an advantage when excavating fossils. Hollow diamond containing filled diamond. Because the fossil is usually formed of a different mineral than the surrounding rock, they can often dissolve the outer rock bed to remove the embedded fossil. Hollow diamond containing filled diamond, for example, silicate replications can be extracted from limestone using an acid. The acid will dissolve the calcareous limestone, leaving the silicate fossil unscathed. Insect fossil record. Hollow diamond containing filled diamond. The insect fossil record has many gaps. Among the primitive apterogoats, only the columbolans, springtails, have been found as fossils in the Devonian period, about 419.2 million to 358.9 million years ago. Hollow diamond containing filled diamond. 10 insect orders are known as fossils, mostly of late Carboniferous and Permian times, 318 million to 251 million years ago. Hollow diamond containing filled diamond. No fossils have yet been found from the late Devonian, about 382.7 million to 358.9 million years ago, or early Carboniferous, about 358.9 million to 318 million years ago period, when the key characters of present-day insects are believed to have evolved. Thus, early evolution must be inferred from the Archodonata, Blatoptera, Coxoplectoptera, Diaphanopterodia, Glosslytrodia, Meganysoptera, Megacycoptera, Myomoptera, Monora. Archodonata. Hollow diamond containing filled diamond. Archodonata is an extinct order of Paleozoic Paleopterous insects, sometimes included in Odonata. Blatoptera. Hollow diamond containing filled diamond. Blatoptera, or proto-cockroaches, is a name given to various, roachid, fossil insects related to cockroaches, termites and mantises. Hollow diamond containing filled diamond. In general, cockroach-like appearance and possibly habit. Hollow diamond containing filled diamond. The fossils assigned to the roachids, or Hollow diamond containing filled diamond. The fossils assigned to the roachids, or of general cockroach-like build, with a large disc-like pronotum covering most of the head, long antennae, legs built for running, flattened body and heavily veined wings. Hollow diamond containing filled diamond. The trait that separate them from modern roachids or external ovipositor, which is a primitive feature in absence of ootheca. Coxoplectoptera. Black small square Coxoplectoptera, or chimera wings, is an extinct order of winged insects containing one family, Mycaladiidae. Black small square Coxoplectopterans are closely related to the mayflies. Black small square 2 adult and more than 20 nymphal fossils have been scientifically described from Mesozoic outcrops. Diaphanoptera. Hollow diamond containing filled diamond. The Diaphanopterodia or Paramegacycoptera are an extinct order of moderate to large size Paleozoic insects. Hollow diamond containing filled diamond. Hollow diamond containing filled diamond. They are first known from the Middle Carboniferous, 60 Maya, and include some of the earliest known flying insects. Hollow diamond containing filled diamond. They have the ability to flex the wings over the abdomen during rest. Hollow diamond containing filled diamond. They are generally considered to be a monophyletic group. About 10 families are known. Hollow diamond containing filled diamond. The group died out at the end of the Permian period, victims of the end Permian mass extinction, without leaving any descendants. Glosslytrodia. Hollow diamond containing filled diamond. Glosslytrodia is an extinct order of insects, containing about 30 species. Hollow diamond containing filled diamond. Its fossil record dates from the Permian to the Upper Jurassic, and is distributed across Eurasia, the Americas, and Australia. Hollow diamond containing filled diamond. Its classification is uncertain, but may be closely related to Neuropterida or Orthoptera. Hollow diamond containing filled diamond. The Glosslytrodia order resonates to the Neuropterida species through the constructional shape of its wings, whilst it is related to the Orthoptera species through the organization of its veins. Meganysoptera. Hollow diamond containing filled diamond. Meganysoptera is an extinct order of very large to gigantic insects, which may informally be called griffin flies. Hollow diamond containing filled diamond. The order was formerly named Protodonata, the Proto-Odonata, for their similar appearance and supposed relation to modern Odonata. Hollow diamond containing filled diamond. Though most were only slightly larger than modern dragonflies, the order includes the largest known insect species, with wingspans of up to 71 centimeters. Hollow diamond containing filled diamond. Most specimens are known from wing fragments only, with only a few as complete wings, and even fewer with body impressions. Hollow diamond containing filled diamond. These show a globose head with large dentate mandibles, strong spiny legs, a large thorax, and long and slender dragonfly-like abdomen. Hollow diamond containing filled diamond. Like true dragonflies, they were presumably predators. Hollow diamond containing filled diamond. Unlike some earlier insects, Megacycopterans bore 2 pairs of wings, which were nearly of the same size. Hollow diamond containing filled diamond. The wings probably were held horizontally, as in dragonflies. Hollow diamond containing filled diamond. The body is usually long and thin. Megacycoptera. Hollow diamond containing filled diamond. Megacycoptera is a Paleozoic insect order. There are 22 known families of Megacycopterans, with about 35 known genera. There are 22 known families of Megacycopterans, with about 35 known genera. Hollow diamond containing filled diamond. They have sucking beak-like mouthparts. Hollow diamond containing filled diamond. Wings are hooded shape with specialized venation. Hollow diamond containing filled diamond. Another distinctive feature was the presence of a number of fine processes projecting from the body, which in some cases could be longer than the body itself, forming long fringes on the insect's underside. Hollow diamond containing filled diamond. They were recorded from Pennsylvanian fossils. Mimotera. Hollow diamond containing filled diamond. Myomoptera is an extinct order of insects, although it is thought to be a common ancestor of all holometabolous insects. Hollow diamond containing filled diamond. Since no smooth transition between Myomoptera and other holometabolous insect orders is known, it is considered to be in a separate order unto itself. Hollow diamond containing filled diamond. The Myomopterans were small insects, with unspecialized chewing mandibles and short abdominal cerci. Hollow diamond containing filled diamond. They had 4 wings of equal size, with a relatively simple venation, similar to that of the more primitive living holometabolous insects. Hollow diamond containing filled diamond. They were existing during early Pennsylvanian to Permian period. Monora. Hollow diamond containing filled diamond. Monora is an extinct suborder of wingless insects in the order Archaeognatha. Hollow diamond containing filled diamond. They resembled their modern relatives, the silverfish, and had a single lengthy filament projecting from the end of the abdomen. Hollow diamond containing filled diamond. They have 10 complete segments with paired stylets in abdomen. Hollow diamond containing filled diamond. The largest specimens reached 30 mm, 1.2 in or more. Hollow diamond containing filled diamond. They belong to Upper Carboniferous and Lower Permian period. Hollow diamond. Gall insects. Plant insect galls are better known than the organisms that produce them. Galls are colorful and unusual deformations of the meristematic growing plant tissues. The organisms are usually small and difficult to identify. One of the first habits of insects observed by man, although most likely unaware of the origin, was that of gall making. Pliny, about 60 AD, describes plant galls and their rapid growth. Malpighi, the Italian physician and botanist, in 1686 attempted an explanation in the De Gallis. It was common knowledge at the time that plant galls had medicinal and curative properties. Theophrastus was the first record in 372 to 286 BC. Although galls are now thought not to have such benefits for humans. Galls are COLOUFUL, unusual deformations, abnormal plant growths of the MERITEMATIC tissues of plants caused insects, mites, nematodes, fungi, bacteria and viruses. Galls can be caused by feeding or egg laying of insects and mites. Definition and features. A plant gall is any abnormal growth caused by another organism, in this case we are speaking of insects, but galls are caused by mites, fungi, viruses, bacteria, and nematodes among others. However, as will be discussed later, the plant interacts with the causative organism and provides the raw materials, proteins, fats, carbohydrates, to construct the gall via abnormal tissue growth. Galls provide a home for the insects, where it can feed, lay eggs and develop. Each type of gall producer is specific to a particular kind of plant. Galls may appear as balls, knobs, lumps, or warts, being characteristic of the causative organism. They appear in various colors like red, green, yellow, black. Factors such as weather, plant susceptibility, pest populations affect the occurrence of galls from year to year. Plant gall damage is usually an aesthetic problem and is not considered serious. Affected trees ordinarily show little injury, although foliage of young trees is sometimes completely deformed. Galls are abnormal growths that occur on leaves, twigs, flowers, fruits, trunks, roots or branches. There are 1,500 species of gall producers, the majority of which are insects and mites. Some galls form where insects or mites feed or lay eggs. They may also develop as a response to infections by several kinds of fungi, bacteria, and viruses. Insects and mites are the most common organisms that cause galls. These organisms can be called horticulturists, for they develop new plant structures. Many leaf miners can produce cell proliferations that integrate into true gall development. The attempt by plants to overcome injury by producing callus tissue is in a sense a kind of gall formation. The family Cystidaemiidae in the order Diptera flies have many species that integrate between gall making and leaf mining. The tulip spot gall, the Codipolosis lyriodendri, is only a slight thickening of the leaf. The tar spot gall, produced by a species in the genus Asteriemia, has even less thickening and is little more than a leaf mine. The boxwood leaf miner, Monothropalpus buxi, makes a leaf mine but the surrounding tissues proliferate. Galls or cecidia are a kind of swelling growth on the external tissues of plants or animals. Plant galls are abnormal outgrowths of plant tissues, similar to benign tumors or warts in animals. Plant galls are often highly organized structures and because of this, the cause of the gall can often be determined without the actual agent being identified. This applies particularly to some insect and mite plant galls. The study of plant galls. Development. Initiation, growth. Maturation. Maturation. The study of this applies particularly to insect galls are the highly distinctive plant structures formed by some herbivorous insects as their own microhabitats. They are plant tissue which is controlled by the insect. Galls act as both the habitat and food source for the maker of the gall. The interior of a gall can contain edible nutritious starch and other tissues. Some galls act as physiologic sinks, concentrating resources in the gall from the surrounding plant parts. Galls may also provide the insect with physical protection from predators. Initiation. Insect galls are usually induced by chemicals injected by the larvae or the adults of the insects into the plants, and possibly mechanical damage. After the galls are formed, the larvae develop inside until fully grown, when they leave. In order to form galls, the insects must take advantage of the time when plant cell division occurs quickly, the growing season, usually spring in temperate climates, but which is extended in the tropics. The meristems, where plant cell division occurs, are the usual sites of galls, though insect galls can be found on other parts of the plant, such as the leaves, stalks, branches, buds, roots, and even flowers and fruits. Gall-inducing insects are usually species-specific and sometimes tissue-specific on the plants they gall. Growth. As it grows, the gall and the insect use nutrients from the host plant. Gall makers may live within individual chambers or within communal chambers inside galls, depending on the species. Maturation. Mature galls stop growing and cease to use host plant nutrients. The developing insect remain protected inside mature galls, grazing on the ready food source. Galls produced by insects and mites include ash flower gall. This gall is caused by a small mite that causes irregular distortion of male flowers. The galls are initially green, then dry and turn brown. Ash midrib gall. Normally 0.5 to 1 inch long, these galls are succulent and have thick walls. A small cavity within each gall contains one or more small maggots, the larval stages of very small flies called midges. Female midges lay their eggs in very young leaflets during early spring. Gall formation begins soon after the eggs are laid. Specifics of the biology of this insect are not known. The galls probably do not harm tree health. Elm coxcomb gall. These distinct galls, caused by an aphid, are about 1 inch long and about 1/4 inch high. The irregular edge of the gall and its red color at maturity account for the common name. The galls dry, harden and turn brown as they age. Aphids may be seen through a slit-like opening in the underside of the gall. This insect has a complex life cycle. It forms galls on elm in early summer, then feeds on grass roots later in the summer. The galls apparently do not cause significant harm to the tree. Hackberry leaf gall. This gall is caused by a small, 0.1 inch long, aphid-like insect with sucking mouthparts called a jumping plant louse. The adults spend the winter under bark crevices and can invade houses in large numbers in the fall. Females lay eggs over a long period of time beginning when leaves begin to unfold from the buds in the spring. Feeding by the nymphs that hatch from these eggs causes abnormal plant growth that forms a pouch. The psyllids remain inside the gallus until they emerge as adults in late summer to early fall. There is one generation each year. Heavy infestations can result in premature leaf drop which over a series of years may affect tree health. Honey locust pod gall. This gall is caused by a small fly midge. The sunburst cultivar appears to be very susceptible to this pest. Infested leaves have globular or pod-like distortions that contain 1 to several small maggots, 0.25 inch long. Infestations begin when females lay eggs in young leaflets. There are 5 or more generations each year. Infested leaves often drop prematurely and repeated damage can kill small branches. New shoots develop at the base of dead twigs. As a result, the natural shape of the tree may be lost. Petiole and stiepule galls. Thick globe-like galls can develop on leaf petioles and stems. Many of these are caused by insects called phylloxerans which are very similar to aphids. The hard, woody galls may remain on the tree for several years. Usually, there is one generation each year. Willow shoot galls. These swellings on shoots, twigs, or leaf petioles may be caused by small flies, midges, or small wasps, sawflies. The gall increases in size as long as the immature stages are active. They cause no significant injury. The infestation may be reduced by pruning and destroying the galled areas before the adult insect emerges, usually in late summer. Witch hazel gall. This gall is caused by an aphid that laid eggs on twigs of the plant. Feeding by the aphid causes the formation of conical galls on the upper side of the leaf. Each gall, produced by a single aphid, later becomes filled with offspring. Mature aphids with wings leave the galls in late spring and early summer and fly to birch. After several generations there, the insects return to witch hazel to lay the eggs that survive the winter. No galls are formed on the birch. Economic importance. Tannic acid is one of the main economic products obtained from galls. A gall made by an Eurasian cynipid yields 65% tannic acid while American sumac galls yields 50%. Dyes are also obtained from galls. Turkey red dye is found in the mad apple gall. Natives of East Africa used to use galls as a source of dye for tattooing. However, galls were apparently not useful to American Indians. Pliny states that the Aleppo gall is able to dye hair black. Inks are also made from some galls, such as the Aleppo gall produced by Cynops galli-tincturii on several species of oaks in Eastern Europe and Western Asia. Laws in some of these regions once stipulated that legal documents be made with inks from this gall. Even the USA Treasury, the Bank of England, the German Chancellery, and the Danish government have specified formulas for inks using the Aleppo gall. Plant galls have also been used in ancient and even fairly recent medicines. Galls were used in the 1700s in France supposedly to control fevers. Today galls are not used medically except in a few ointments. Galls sometimes have been used as food, or rather as a spice. In the Near East, a gall on Salvia pomifera produced by a species of aulex is known as the gall of sage or palm. It is aromatic and acidic in flavor and is used with honey for cooking. Most galls taste similar to the host plant. Some of the galls have a higher content of carbohydrate, as will be shown later for pinion needle galls. Most plant galls, however, are injurious to crops, for example, the Hessian fly, clover leaf midge, chrysanthemum midge, and pear leaf blister mite. Types of galls. There are basically 2 types of galls, open and closed. The open galls are produced by piercing mouthpart forms like aphids, psyllids, coccids and mites. For example, the pear leaf blister mite Areophys pyri, the witch hazel cone gall maker Hormophis hammomelodus, and the elm coxcomb gall maker Calpha ulmicola. They feed on the leaf surface which later folds and grows inward to produce a pocket more or less. Reproduction occurs within the galls and the young leaf through a small opening on the lower surface of leaf. The life cycle of aphids of open galls of witch hazel cone gall is like that of other aphids with alternation of generations between parthenogenetic wingless ones and sexual winged adults. However, the aphids are modified in forms so they hardly resemble free-living forms one usually see on plants. The witch hazel cone gall maker produces sexual winged adults when the gall becomes crowded and these fly to birch, where they have several generations. These aphids are all females and have white wax oozing from wax pores along the margin of the insect. After several generations on birch, winged females return to witch hazel where they produce wingless males and females that mate and lay eggs to overwinter on witch hazel. Closed galls are made by larvae with mandibles such as Coleoptera, Lepidoptera, Diptera, and Hymenoptera. None mate and produce young in the galls. Most galls are simple monothalamus and contain a single larva. Fewer galls are compound polythalamus and have several larvae in separate chambers. Galls can also be classified according to their form and texture. Blister or spot galls are Spangle galls have more thickness and resemble shallow plates on lower surfaces of leaves. Cystidiomyidae, sometimes Cynippidae. Button galls are made by Cynippidae. Tube galls on grape and hickory are made by Cystidiomyidae. Oak bullet galls and oak, apples, are produced by Cynippidae. Irregular woody galls on rose, oak, and blackberry are Cynippidae. Galls on flower heads are made by Cystidiomyidae, sometimes by mites or Cynippidae. Egg scars from tree crickets and leaf hoppers can become calloused and resemble galls. Gall making habit. The gall making habit has been evolved independently in the different insect and mite groups. The gall insect lays its egg upon the host plant or within its tissues. The egg hatches to a small larva which must contact the meristematic cells of the cambium or growing parts. At this point the gall begins to grow. Galls are the result of cell multiplication, hyperplasia, and quite often also the cells are enlarged, hypertrophy. Galls are structures that form as a result of the abnormal growth activities of plants in response to gall-inducing organisms. Most galls are caused by nematodes, insects and mites, while a very small percentage are caused by bacteria, fungi and viruses. There are thousands of species of insects in the world that induce gall formation on the roots, stems, leaves, buds, flowers and fruits of plants in a wide variety of plant families. Insect galls range in complexity from simple outgrowths to more highly differentiated structures such as those typified by many of the cynipid wasp galls. Despite the large numbers and types of insect-induced galls, very little is known regarding the underlying mechanism or mechanisms of insect gall formation. In contrast, the mechanism of crown gall formation by the bacterium Agrobacterium tumefaciens has been well characterized. An understanding of the mechanism of crown gall formation may provide some clues to the mechanism or mechanisms of insect. The gall-making habit is found in 6 orders of insects: Coleoptera beetles, Lepidoptera, moths and butterflies, Homoptera, aphids, Thysanoptera, Thripsdiptera flies, and Hymenoptera, sawflies and wasps. The gall serves both as a shelter and a food source. Also, the gall insect is partially protected from parasites and predators, especially general ones. The inner walls of galls are moist or liquid and are usually rich in protein and sugars, much more than the surrounding non-gall tissues. Many inquilines live in insect galls as associates in this microecosystem. There are also parasites and predators of the gall insects and other associated organisms. For example, Hendel listed 31 dwellers in addition to 1 species of gall maker, 10 inquilines, 16 parasites and 5 brief visiting species. Damage and host plant. Gall-making insects are generally not considered as pests. Some galls are even considered attractive and reused in flower arrangements and other crafts. Most gall-making insects do not damage the host plant, however certain species may cause aesthetic damage such as leaf discoloration, early defoliation, twig, stem drop of valuable plants. Heavy infestation may reduce yield. Host plant type of gall classification: pest. Apple gall on root, twigs, woody apple, aphid, cypress, swelling on growing tips, midge gall, Taxodiomya sp, elm bladder, finger type, leaf gall, mite, Areophys sp, ficus, leaf folding and rolling, thrips, grape gall on roots, blister bud gall. Phylloxera, Pachycyla sp. Oaks leaf galls with orangish hair, sticky, spongy galls on twigs with seed-like structure inside. Gouty oak and horned oak gall wasps. Willow cone-like galls on terminal willow cone gall fly. Gall insects. Leaf miner. Leaf miners are insects that feed within a leaf, producing large blotches or meandering tunnels. Although leaf miner injuries are conspicuous, most leaf miners produce injuries that have little effect on plant health. Insecticides applied when leaf miners lay eggs are useful for control of many leaf miners. Leaf miners are insects that have a habit of feeding within leaves or needles, producing tunneling injuries. Several kinds of insects have developed this habit, including larvae of moths, Lepidoptera, beetles, Coleoptera, sawflies, Hymenoptera, and flies, Diptera. Most of these insects feed for their entire larval period within the leaf. Some will also pupate within the leaf mine, while others have larvae that cut their way out when full-grown to pupate in the soil. Leaf miners are protected from many predators and plant defenses by feeding within the tissues of the leaves, selectively eating only the layers that have the least amount of cellulose. Feeding and tunneling is diagnostic of the species involved. The mine often contains frass or droppings, and the pattern of frass deposition, mine shape, and host plant identity are useful to determine the species and instar of the leaf miner. A few mining insects feed in other parts of a plant, such as the surface of fruits. They are major insect pests of many crop and ornamental plants. Leaf miners tunnel their way through a wide range of plant species. With over 300 species of leaf miners worldwide, there are 5 considered to be serious horticultural pests. Vegetable leaf miner, Liriomyza sativea. Tomato leaf miner, Liriomyza bryonii. Chickpea leaf miner, Liriomyza cicerina. Serpentine leaf miner, Liriomyza wedobrensis. American serpentine leaf miner, Liriomyza trifolii. Areas mined by insects die and dry out. Although injuries produced by leaf mining insects can be unattractive, it is rare for them to significantly affect plant health. Also, most leaf miners have important natural controls which normally check populations before too much injury is done to plants. Adult leaf miners are small, grayish-black flies. They have a yellow and black body and are 1.3–2.3 millimeters in length. The different species vary in color, which makes them identifiable. Adult females are larger than males and more robust. The larval stage of the leaf miner is not usually seen as they remain inside the leaf tissue. Egg-laying punctures can be seen as white speckles on leaves. The feeding track is white, often with an obvious trail of dark waste material inside. Damage caused by the leaf miner reduces the growth and development of seedlings and young plants, which can lead to death. Leaf miners are sometimes classified by the pattern of the mine which they create. Serpentine leaf mines wind snake-like across the leaf, gradually widening as the insect grows. More common are various blotch leaf mines, which are generally irregularly rounded. One subgroup of these are the tentiform leaf miners, which produce bulging blotch-type mines that curve upwards somewhat like a tent as the damaged leaf tissue dry. Mining patterns often are combinations of the above, such as species that initially produce serpentine mines but terminate by making the enlarged leaf cavity of a blotch mine. Serpentine mines. The elongated bending mines are usually called serpentine mines. These are made when the larva advances into one direction during its feeding. The width, length and the widening rate of the serpentine mine are usually typical for the species and can help in the identification. The location of the mined leaf margin, inside the leaf, is also important character. While females of some species lay eggs on the leaf margins, others do in the middle of the leaf blade, so the mine begins. Respectively, the serpentine mines often follow the veins, and in some cases it is coiled in a spiral line. Blotch mines. Blotch mine is made when the larva turns around itself during its feeding. So the width and length of the mine is not as different as in case of serpentine mines. The shape of blotch mine can either be totally irregular or quite regular, i.e. circle, oval. Many species of lithocolitid leaf miners, family Lithocolitidae, make typical oval blotch mine. Other species, i.e. Parectopa robiniella, makes a regularly forking, amoeba-like, blotch mine. The family Cystidaemiidae in the order Diptera, flies, have many species that intergrade between gall-making and leaf mining. Some leaf surface feeding insects may appear like leaf mines. Some Chironomidae Diptera dig grooves in leaves, removing the upper epidermis and packing the channels with frass. In the beetle order, there are 50 North American species, scattered among 3 families, Buprestid, Chrysomelidae, and Curculionidae. The Buprestid are the metallic wood-boring beetles, are flat-headed borers. Most larvae bore under bark or in wood. Adults are metallic copper, green, blue, or black with hard bodies and can move rapidly with good eyes. The mines of Buprestid are usually of the blotch type, a cave or irregular area, but some make wide linear mines. The genus Brachys have species that mine leaves of oak, and these species when disturbed draw up their legs and play dead, usually falling to the ground to avoid predation. Other leaf mining genera are Trachys and Pachycelis. These beetles often visit flowers as adults. The Chrysomelidae leaf beetles is one of the largest families of beetles and includes the Colorado potato beetle. These beetles are relatively small, about 1 centimeter or less, and often brightly colored. There are many serious pests in this family, and depending on the species, the larvae feed externally on leaves or roots, bore into the plant, or are leaf miners. The elytra wing covers often are spiny or may be ridged and sculptured. The larvae are flattened, short legs, and closely resemble the Chrysomelidae. Another subfamily, Halticinae, has Dibelia borealis, which makes long winding mines on the broad-leaved plantain which may wither the plant. Hippuriphylla. Modiri makes blotch mines on leaves of Rumex. Diptera. Agromyzidae. Anthemiidae. Hymenopterans. Tenthredinidae. The Tenthredinidae, common sawflies, is a large group with about 9 out of 10 sawflies usually seen. Adults are wasp-like, often brightly colored, on leaves or flowers, medium-sized to small of 20 millimeters or shorter. Some species are gall formers, Yura on willow. The birch leaf miner, Fenusa pusilla, makes blotch mines and can be a serious pest. The elm leaf miner, Fenusa ulmi, also can be damaging. The two sexes are often differently colored. The leaf mining species have reduced legs and all make blotch mines. Lepidoptera. Nepticulae. Tischariidae. Gracilariidae. Galechiidae. Moths, some only with wingspread of 3 mm, wing venation reduced, surface of wings have spine-like hairs. Most species make linear leaf mines and pupate in soil. Some Ectodermia are gall makers. Tischariidae, small moths, hind wings are long and narrow, small or absent maxillary palps, make blotch mines in oak, apple, blackberry and raspberry. Apple leaf trumpet miner Tischaria mellifoliella is common in eastern USA. They make trumpet-shaped mine in upper surface of leaf. Gracilariidae, leaf blotch miners, a large group of small to minute moths with pointed wings, often fold leaves. The white oak leaf miner Lithocolitis. Hamadryadella is common in eastern USA and feeds on oaks. Many mines, one larva to a mine, may occur on a leaf. Larvae are flattened with rudimentary legs and prothoracic segment enlarged laterally, pupate in mine. Leaf mining can have economic importance when succulent leaves, such as parsley and delphinium, wilt as a result of the mining. The leaves of woody plants usually remain turgid and functional in spite of the damage. The leaves of vegetables, fruit, or ornamental plants are often damaged the most severely by mining. Spinach and beet are mined by Pegomya hyoscyae. In the past, chemical sprays have been rather ineffective against leaf miners due to their protection within the plant. Ecological aspects. Eggs laid on leaf surface. Larva cut into leaves or adult female cut hole in leaves and deposit the eggs. Larva feeds for few instars. Head wedge shaped to cut the epidermal layers. Small legs, antennae and eyes are also reduced. Food consists of palisade cells, parenchyma and oozing sap. If latex cells cut, it may drown and kill the larva. Veins may impede their movement. Frass deposits often reduces its saps. Either it is removed or larva moves to another leaf. Leaf mining during end of summer. If it happens during spring, they turn to galls. Common leafminers of trees and shrubs. Sawfly leaf miners. Most sawflies chew on the surface of leaves, but 4 species found in Colorado develop as leaf miners of woody plants. Adults are small, dark-colored, non-stinging wasps that insert eggs into the newly formed leaves. The developing larvae produce large blotch mines in leaves during late spring. The sawfly leaf miners produced a single generation each year. Ulmi is the most important species, being locally common in several Front Range cities where it develops on American, English and Siberian elms. Other species include hawthorn leaf miner Prophanusa canadensis associated with Crataegus cruscalli, C. persimilis, and C. erectus, birch leaf miner Phanusa pacilla present in some plantings of white or gray birches, and alder leaf miner Phanusa dorni, a native species that develops in alder leaves. Tentiform leaf miners. Larvae of several tiny moths Philoneuricter species produce blotch mines in leaves that pucker when they dry out, somewhat resembling a pup tent. These tentiform leaf miners occur on willows, poplars, and cottonwoods. Hackberry and apple and leaf mines tend to be concentrated on the lower, shaded leaves. Probably 2 generations are normally produced. Outbreaks are rare because these insects are normally heavily attacked by parasites and other natural enemies. Lilac miner. Another small moth, the lilac leaf miner Coloptilia syringula produces a blotch mine and then folds edge of lilac. A related species, the box elder leaf miner, Coloptilia negandella, produces similar leaf injuries to box elder leaves. There are 2 generations per year and the life cycle is likely similar to that of the lilac leaf miner. Needle miners. Several minute moths of the genus Coleotechnites have larvae that develop in the needles of conifers. Affected needles appear brown beyond the tunnels of the larva. Most important is the ponderosa pine needle miner Coleotechnites ponderosae that has periodically produced outbreaks in forested areas of ponderosa pine. Related species occur in lodgepole pine, pinyon, and spruce. Aspen leaf miner. Delicate, winding mines through the upper leaf surface of aspen leaves are characteristic of another small moth, Phyllochnistis populiella. The feeding by the caterpillars that produce the mines produces insignificant damage and is notably mostly because they may attract attention. Poplar black mine beetle. Large black blotches in poplar leaves are produced by larvae of a leaf beetle, Zoocophorus scutellaris. Adults chew small pits in the leaf. Outbreaks are extremely rare and the insect causes only minor leaf damage. European elm flea weevil. Probably the newest leaf mining insect to establish in Colorado is the European elm flea weevil, Orches salmi. The larvae make a leaf mine in elm that superficially resembles that of the elm leaf miner sawfly. The mine of the European elm flea weevil originates from a leaf vein, meanders in a serpentine form, and ultimately terminates in a leaf blotch along the edge of the leaf. Adults are tiny beetles that jump which makes pits in leaves, producing a lacy appearance of foliage when they are abundant. Columbine leaf miners. At least 2 species of leaf mining flies in the genus Phytomesa produce leaf mines in columbine. One produces blotch mines while serpentine mines are characteristic of the second species. Adults are tiny dark flies and females make leaf punctures with their ovipositor so that they can drink plant fluids. The presence of small puncture marks is an indication of activity of these insects and can be useful for timing insecticides applied for control. Vegetable leaf miners. Several Liriomesa species of leaf mining flies make long serpentine mines through leaves of flower and vegetable garden plants. Normally these insects are very well controlled by natural enemies and outbreaks are almost always associated with use of insecticides. Spinach leaf miners. The only insect that regularly mines edible parts of plants is the spinach leaf miner Pegamia hyoscyame, which produces large, dark blotch mines in leaves of spinach, beets, and related weeds. Adults are small gray flies, about half the size of a housefly, and they emerge in spring to lay eggs on the underside of leaves. The eggs of this insect are quite distinctive, being white and laid in small masses. Problems are most common in gardens where spinach and beets are overwintered and continuously grown, providing host plants for the insects. Injuries most commonly occur in spring, but there are 2 or more generations produced during the growing season. Controls of leaf miners of trees and shrubs. Few, if any, leaf miners pose any significant threat to the health of trees or shrubs. Injuries are cosmetic and treatment decisions are based on plant appearance. Furthermore, most leaf miners have numerous natural enemies that normally well regulate their populations. Outbreaks, if they occur, are usually very short-lived. However, exceptions to this can occur among the non-native species that are not as well regulated by natural enemies in areas such as Colorado where they have been recently introduced. Examples of non-native leaf miners are those found on elm leaf miner, European elm flea weevil birch leaf miner, and the tentiform leaf miners of apple. If controls are attempted with insecticides, there are 2 approaches to consider. The first involves sprays applied to leaves that are timed to coincide with periods when egg laying occurs, usually shortly after leaves have expanded in spring. Persisting contact insecticides can be used for this purpose, such as the various pyrethroids that currently predominate for control of insects that chew on leaves: permethrin, bifenthrin, lambda-cyhalothrin, cyfluthrin, deltamethrin. None of these will move systemically in plants, so active larvae within mines will not be affected. Sprays of systemic neonicotinoid insecticides. Imidacloprid, dinotefuran can provide some control of these stages among leaf miners that are beetles Coleoptera or sawflies Hymenoptera. Neonicotinoids tend to be poor in control of caterpillars, the larval stage of moths Lepidoptera. Alternately, neonicotinoid insecticides can be applied to the soil for root uptake. Imidacloprid merit, various generics, is widely available for this application both through most nursery outlets and by commercial applicators. Dinotefuran Safari is presently only available through commercial applicators. Controls have not been evaluated for the columbine leaf miners, but they are likely able to be managed in the manner of leaf miners on trees and shrubs. Furthermore, the feeding puncture wounds that the flies make can provide an early means of detecting their activity, thus allowing proper timing of treatments applied against the egg-laying adults. No controls for the vegetable leaf miners are recommended. Many insecticides have poor activity against these insects, and often insecticide applications will make problems worse by differentially destroying natural enemies. Spinach leaf miners pose different problems as they appear on edible crops. In gardens, the most simplest and most effective means of controlling this insect is to regularly check the plants for the presence of eggs, which can be hand crushed. Leaves with actively growing larvae should be picked and destroyed. Leaving picked leaves on the ground will allow the larvae to finish development. The key aspect of managing leaf miner infestations is to detect them before it becomes to deal with. Plant inspection is one detection method. Another method is to use yellow sticky traps, placed slightly above crop height. Pyrethrins, which are derived from the flower of Chrysanthemums are effective against some species of leaf miner insects as well as many other garden pests. Pyrethrins are eco-friendly, however they need to be frequently reapplied as they degrade approximately 24 hours after being sprayed. Neem oil. Its effectiveness is based on several modes of action. It disrupts insects' hormonal balance so they die before they molt to the next life stage. It suppresses some insects' desire to feed and also repels in areas that have been sprayed. Leaf miner. Egg. Filled circle, usually ovoid. In the case of parasitic forms, eggs have a pedicel, the function of which is supposed to pierce the body wall of the host and allow the newly hatched larva to breathe outside air. Larva. Filled circle symphyta larvae are with well-developed head, 3 thoracic and 9 to 10 abdominal segments. Filled circle tracheal system is peripneistic or holopneistic. Filled circle 3 pairs of thoracic and 6 to 8 abdominal limbs are present. Filled circle apocrita larvae have less chitinized head, they are opodous and in parasitic forms, it is sunken into the prothorax. Filled circle in parasitic forms, degeneration of sense organs is seen as they live in darkness and provided with ample nutrients in their vicinity. Filled circle mandibles are dentate and sickle-shaped, maxillae, labrum and labium are fleshy lobes. Filled circle, the stomach is a blind sac, not communicating with the hind intestine, and the fecal matters are expelled only at the conclusion of larval stage. Filled circle, well-developed salivary glands are seen in phytophagus forms, ventral nerve cord ganglia are undifferentiated, and the tracheal system is holopneistic. Filled circle in parasitica, the respiratory system is modified according to the environment. Ectoparasites are peripneistic and endoparasites are apneustic. Filled circle hypermetamorphosis is seen in parasitica with different type of larvae. 1. Planidium, active with chitinized segments, and spine-like locomotory processes. Egg is laid away from the host and the larva migrates towards the host. E.g., Chalcidoidea. 2. Caudate, vermiform with caudal outgrowths. E.g., Ichneumonidae. 3. Cyclopid naupliform, large swollen prothorax, sickle-shaped mandibles and bifurcated caudal processes. 4. Teleform, cephalic extremity is hooked, posteriorly body is prolonged to a caudal process, trunk is provided with setae. 5. Vesicle bearing, proctidium is everted to from swollen anal vesicle. 6. Eucoiliform, cephalic process and setae are absent, 3 pairs of thoracic appendages seen. 7. Polypod, 8 to 12 pairs of trunk appendages, later undergoes hypermetamorphosis and final instar is maggot-like. Pupa. Filled circle, there is a prepupa between larva and pupa, the prothoracic segment is distended, wings and legs are pupa-like, propodium is seen. After the final larval instar, an exarate pupa is formed. A coccon is generally seen. It can be a silken type as in Aculeata or formed of soil particles as in Tenthredinoidea. Filled circle Hymenoptera has 2 suborders, Symphyta and Apocrita. Filled circle Apocrita has 2 subdivisions, Aculeata stinging forms and Parasitica parasites of other insects. Symphyta. Filled circle primitive members of Hymenoptera are included in this group. Filled circle abdomen has no constriction, trochanters 2-jointed, larva with thoracic and abdominal legs. Filled circle, a single pair of ocelli maxillary palp 4-jointed and labial palp 3-jointed. Filled circle ovipositor adapted for sawing. Filled circle, the larvae have well-developed head and 13 trunk segments, 3 pairs of thoracic legs and 6 or more pairs of abdominal legs. Filled circle, it has 2 superfamilies, Tenthredinoidea, Oriswadea. Apocrita. Filled circle abdomen separated from thorax by a deep constriction or petiole. Filled circle, they show social organization. Filled circle trochanter 1 or 2-jointed, larva opodous. Filled circle larval habits vary widely. Some are phytophagus Chalcidoidea, some are predaceous Vescoidea, some are ecto or endoparasites Ichneumonidae, and some are nectar-feeding Apidae. Apocrita. Superfamily Ichneumonidae. Filled circle antennae not elbowed, no angle between 2 filaments, pronotum extending back to the tegulae, small scale-like sclerites of the mesothorax, trochanter 2-jointed, forewing with a stigma, ventral segments of abdomen membranous, ovipositor issuing some distance before the apex of the abdomen. Filled circle, they are all parasites on other insects and hence beneficial to man in pest control. Family of Oniidae. Filled circle, the abdomen is highly compressed with the base inserted into the dorsal side of metathorax. Filled circle, a costal cell separates costal and subcostal veins. Filled circle, Ivania appendigaster is a parasite of Periplaneta, Olicus is parasitic upon larva of Cerambicity. Family Ichneumonidae, ichneumon flies. Filled circle, they are parasites and hyperparasites of Lepidoptera and Coleoptera. Some parasitize Arachnida and some attack Aphididae. Filled circle, they are the most highly evolved solitary insects. The adults are active during sunny days and prefer flowers. Filled circle, females hibernate but males perish before winter. Filled circle, body is large, slender, black, yellow or brown in color. Filled circle, antennae filiform, long with 16 segments. Filled circle, hind femur with a trochantellus segment between trochanter and femur. Filled circle, forewings have pterostigma at distal end of costa, venation complete. Pterostigma at distal end of costa, venation complete. Filled circle, gaster bulbous posterior portion of the metasoma 3 times longer than head. Filled circle, apteris and brahipterous forms are found in Cryptinae. Filled circle larvae have variably shaped head and 13 body segments. Filled circle 9 pairs of spiracles, first on thorax and 8 on abdomen. Filled circle in endoparasitic species. The caudal filament acts as a respiratory organ, respiratory system being apneustic. The caudal tail disappears in the third instar and tracheal system becomes functional. Filled circle in ectophagous forms, respiratory system is peripneustic. Filled circle when well fed, the larva spin cocoons of iridescent yellow, white or black threads. Filled circle. The most remarkable group in the family is the genus Thalassa, whose ovipositor is several inches long. It is used to drill the wood of trees to reach its host and lay eggs in the host body. Family Braconidae. Filled circle. They resemble Ichneumonidae except that third discoidal and second apical cells of the wing are confluent. Filled circle. Filled circle. They commonly parasitize Lepidoptera. Filled circle larvae have chitinized large head, 13 body segments, 9 pairs of spiracles, first pair on the mesothorax and 8 on abdomen. Filled circle endoparasitic larvae have caudal appendage. Filled circle pupation can occur either in the host body or outside, many larvae found in the body of a single host. Filled circle pupa is enclosed in a silken white, yellow or buff-colored cocoon. Filled circle members of a species emerging from the same host construct their cocoons in a mass arranged in the form of a honeycomb. Filled circle Aphidiinae parasitizes on Aphididae, only a single larva developing in the host body. If the aphids are attacked before the second ecdysis, the host cannot attain maturity. If the aphid is attacked after third instar, it still can give rise to small number of progeny before succumbing. Filled circle when about to pupate, the larva makes a ventral fissure in the host tissue and cements the host body on the substratum over which it pupates. Finally, the adult emerges through a hole. Superfamily Chalcidoidea. Filled circle antennae not elbowed, pronotum not extending to the tegulae, trochanters 2 jointed, wings without stigma. Filled circle. It is the highest with 29 morphologically different forms discovered. 1. Male anert sense organs, wings and genitalia are well developed, mandibles are less developed. Head is smaller and rounder than females and workers, antennae are more long and slender too. Female giant, the queen is large with well-developed reproductive organs. Longer than male and worker. Antennae and legs are shorter and stouter than male. Mandibles are well developed and gaster is larger. 3. Gaster or gate, wings absent, reduced thorax and small gaster, ewes are small, ocelli small or absent, ovarioles greatly diminished and seminal receptacle is wanting. Antennae, mandibles and legs are well developed, can be of various sizes. 4. When the number of any of the former casts assume large size, they are called macroner, macrogyne and macroergate. 5. When the casts are dwarf-like, they are called microner, microgyne and microergate. 6. When the larvae of some tropical ants are parasitized, they fail to attain the imago stage, wings are suppressed, many parts are absent and they are then called thesaner, thesogyne and thysergate. 7. When individuals are attacked by nematode myrmis, the wings are reduced, workers have enlarged body and develop female characters, then they called myrmithaner, myrmithogyne and myrmithergate. 8. Sometimes, males and females resemble workers and become devoid of wings, then they are called ergotaner, ergotogyne. 9. An unusually large male occurs in Doridinae, with large mandibles, long cylindrical abdomen, singular genitalia, then it is called dorolaner. 10. In parasitic workerless genera of anergates, sometimes males resemble a female, then it is called gynecaner. 11. When male and female characters are found in the same individual, it is called gynandromorph. When a worker character is seen in a male, it is called ergotandromorph. 12. When a female shows excessive development of antennae and wings, it is called a beta female. The normal female is then called alpha female. 13. An aptrous worker-like individual with size and gaster of a worker and thorax of a female, it is called pseudogyne. 14. When a worker starts laying eggs, it is called a gynecoid. This happens when the queen is lost and the workers are well fed. The eyes and wings are absent with enlarged gaster and ovaries. 15. Soldier dinergate, large head and mandibles adapted for fighting, guarding the nest, crushing hard food particles, etc. 16. Desmergate is an intermediate between a worker and a dinergate. 17. Pyrorgate replete, a worker which distends the abdomen with stored liquid food until it becomes a large sac and interferes with locomotion. 18. Pterogate, worker or dinergate with vesigal wings and unmodified thorax. Colony formation. Filled circle. When both sexes are winged, mating takes place during nuptial flight. Filled circle workers will not allow male and female to come out of the nest until the climatic conditions are suitable. Filled circle. After mating, the female discards the wing and excavates a hole in the ground. Filled circle. The female remains in the chamber till the eggs are mature enough to be laid. Filled circle. During this period, the female depends on the fat reserves of the body as well as the nutrients released from the histolysis of the wing muscles. Filled circle. When the first larvae appear, they are fed by the female's salivary gland secretions till they pupate. Filled circle. When the workers are full grown, they come out and establishes contact between brood chamber and the colony. Filled circle. Then they forage for food and share it with the exhausted parent. Filled circle. Now the female feeds on the liquid diet provided by the worker and dedicates herself to only laying the eggs for up to 15 years. Filled circle. The number of individuals in a colony vary between 20,000 to even 500,000. When the colony becomes excessive, it splits and each with a few workers and one queen migrate to a new place. Nest formicaries. Filled circle. Different species have different architecture and even modifications are carried out according to the conditions. 1. Harvester ants construct sandy mound with water drainage system. Workers bring eggs, larvae and pupae into the sun in the morning and return to the nest at noon. E.g., Formica rufa. 2. Leafcutter ants construct massive nests of 20 feet in diameter descending to a depth of 10 feet underground with many openings and chambers connected with tunnels. They also grow fungal gardens. E.g., Trachymermis. 3. Arboreal ants secrete silken threads which are woven into the nest using mandibles. Some species develop colonies in some hollow twigs or thorns. The nests sometimes hang from the tree which contains galleries and chambers. E.g., Oecophila 5. Some species lack nests. For egg laying, a temporary nest called bivouac is constructed by weaving a silken thread which remains intact for 24 hours. E.g., army ant 6. Some species don't build nests but live below stones or logs or in the nests of other ants. Both queen and workers are blind. E.g., Doralinae. Food and feeding. Filled circle ants feed on a wide range of food. They can be herbivores, carnivores, Doralinae, or scavengers. Filled circle. The harvesting habit evolved as a solution to increasing competition for food, especially in dry arid regions. E.g., Myrmecinae. Filled circle. Some feed on fungi and cultivate fungal garden in the nest. Filled circle. The queen build the fungal garden. Before nuptial flight, the queen fills her infrabuccal chamber with fungal hyphae and sprinkle it on the newly formed nest chamber. The hyphae are nourished with the insect's fecal matter and even with some of the eggs. Filled circle. Fungus garden is a sponge-like structure developed on leaf pieces and insect excrement. Fungus grows rapidly and produces numerous swellings which form the food for larvae and adults. Filled circle. Ants show trophallaxis. Ant larvae are fed by workers and in return, larvae secrete a fluid from dermal glands which the adults ingest. Filled circle. While collecting food, the workers release pheromone which initiates trails so that they move in a line one after another to search for food and transfer into the nest. Filled circle. The subfamily Formicinae is called honey ant. Filled circle. Here the workers have integument which can be inflated to store the nectar collected. Such workers are called repletes. They, on returning to the nest, regurgitate the nectar. Filled circle. In the nest there are other repletes, which receive the nectar brought by the foraging repletes. They regurgitate this nectar to feed the larva. Thus they act as living bottles. Social associations. Filled circle ants have association with phytophagous insects belonging to the group Aphididae, Coccidae etc. for feeding the liquid secreted by them. Filled circle ants offer protection in return by building tents or shelters around them, so it is a kind of symbiosis. Filled circle. When the nest is disturbed, ants carry the aphids and even their eggs to a place of safety, often carrying the nymphs to a stem or twig close to the nest. Filled circle. Some ants show slave-making behavior. Filled circle. Queen enters the nest of another species of ant, captures a portion of the brood. The slave brood follows the queen and feeds the larva till the workers of the encroacher start brood supply. Filled circle. Sometimes workers capture larvae from the colony of other species of ants and bring them to their colony. These larvae, when become workers, will feed the new colony. By that time the original workers of the colony will either become blind or inactive. Filled circle. Some organisms inhabit the ant's nest either throughout the life or at some stages. They are called myrmecophiles, ant guests. E.g., Coleoptera. Filled circle. The relations of myrmecophiles are as follows. 1. Synecthrans. Scavengers or predators treated with hostility. They feed on dead or weak ants, refuse etc. They have to elude the ants to get food. 2. Synsecates. Indifferently tolerated without inviting the attention of ants due to their small size or absence of any odor. Sometimes they get food by imbibing the food regurgitated by the worker ants or lick the ants to get cutaneous secretion. 3. Symphiles. True guests, amicably treated, fed and even reared by ants. The ants lick the body of the guests to feed on their dermal secretions. 4. Parasites. Are the parasitic Hymenoptera and nematodes. Some ants also show social parasitism. The queen enters the nest of another species, kills the queen and causes the workers of that colony to rear her first brood. Gradually the workers of that colony die and the nest is entirely peopled by the parasitic ants. 5. In some cases, social symbiosis is shown among different species of ants where they share a common nest and live amicably but keep their broods separate. Usually one of the partners will be smaller in size and feed on the remnants of food collected by the larger partner. Superfamily Vespoidea. Filled circle. Pronotum extending back to the tegula. Colony founding. Filled circle. The female wasps, after hibernation, become active in the spring, seek out for a suitable nesting place and then start collecting materials for nest building. Filled circle. Using mandibles, parts of wood are scraped off, and using saliva, a substance called wasp paper, is produced. Filled circle. A cavity is made in the ground and the wasp paper is applied to the roof, forming a disc, from the center of which a pedicel is hung which broadens to the tip. Filled circle. Upon this pedicel, about 30 hexagonal cells are built, closed above and open below. Filled circle. An umbrella-like covering is placed over the comb and in each cell close to the center an egg is laid, which is fixed using a cement-like substance. Filled circle depending on temperature. In a few days, larvae hatch and are fed by the parent till they pupate. Before pupation, larva spins a silken thread using which the mouth of the cell is closed. Filled circle, the contents of the abdomen are removed for the first time and pupation begins. Filled circle, after 5 to 6 weeks from egg laying, the adults bore through the floor of the cell and emerge. These are the workers. Further care for the larva and nest building are carried out by the workers, and parent now only lays eggs. Filled circle, new layers of cells are built upon the old ones and are connected together by means of vertical pillars. Many such layers are formed and finally the nest looks like a spherical structure from outside. It is well protected from rain and also maintains the temperature inside. Filled circle, near to summer, larger cells are built called the royal cells for the queen of next generation. Filled circle, in a single nest, there will be 7 or more combs and about 5,000 individuals. Filled circle, when food is more or when the colony needs a new queen, the larva will be developed into females, one of which will become the queen. Filled circle, in the nest, other insects may be there which act as either scavengers or parasites of larvae. Superfamily Apoidea. Filled circle, social and solitary bees are included here. Social bees found in the families caste system. Filled circle hive consists of 3 types of bees: queen, worker and drone queen, only fertile female in the hive. She develops from a fertilized egg. Filled circle, queen is the largest member of the colony. Filled circle, the abdomen of the queen is long, extending beyond the wings. Thorax is larger. Pollen basket and wax glands. Sting is curved and smooth. Filled circle, queen does not perform any duty except egg laying. Queen survives for 3 to 5 years and lays around 2,000 eggs per day. Filled circle, queen produces queen pheromone from the mandibular gland, which acts as the social glue that gives the colony its identity and temperament. The workers are kept under control and maintained sterile by queen pheromone. Also, it attracts the drones. They are males developing parthenogenetically. Filled circle, male is larger and stouter than the worker, easily distinguished by the large holoptic eyes and correspondingly reduced frontal parts of the head. Filled circle, they don't have sting, pollen basket or wax glands. Filled circle, they live only to mate with the queen and dies. Workers, they are sterile females developed from fertilized eggs. Filled circle, they build hive, guard the colony, go out foraging, nurse the larva and tend to the queen. Filled circle, they have some special glands. Hypodermal glands secreting wax. Pharyngeal glands to secrete royal jelly. Scent glands to release pheromones to locate food and mark the territory. Pollen baskets to collect pollen. Barbed sting to drive away enemies. Filled circle, worker bees progress through various growth stages. Nurse bees. When they first hatch, they become nurse bees. They clean cells, keep the brood warm and feed the larva. They also receive the nectar from field bees. Guard bees. Keep unwanted visitors away from the hive. Fan to cool the hive. Fan to dehydrate honey and produce locator pheromones from the Nasonop's gland. Undertaker bees. Remove the dead bees. Forager bees. Last stage of life. Gathers nectar and pollen. Acts as pollinators. The lifespan is less due to attack from predators and poisoning from insecticides. Laying worker. They are sterile females, but when the colony becomes queenless, they develop their ovaries and begin to lay unfertilized eggs. Honeycomb. Filled circle, composed of hexagonal cells arranged in 2 series placed back to back. Separate combs hang vertically downwards. Filled circle, the young workers have hypodermal glands on the 4th to 7th abdominal sterna, which secrete wax using which honeycomb is constructed. Filled circle, wax is secreted as a liquid from fine cuticular pores which the hardens to form plates, which project from the projects between the adjacent sterna. Filled circle, bee impales the plate on the spine at the distal end of the first tarsal joint of the hind leg. Leg is flexed forward and the plates removed by mandibles and kneaded to required structure to build the cells. Filled circle, the largest cell is the royal cell to rear the queen, followed by the cells to rear the drones. Workers are reared in the smallest cells. Other cells are used to store pollen and honey. Filled circle, along with wax, bees also use a resinous substance collected from the trees to build the comb. This substance is the propolis, which is used as a glue to hold the cells together and fill the crevices. Filled circle, the queen lays a single egg in each brood cell. Larvae hatch in 3 days. When larvae are full grown, workers seal the mouth of the cell with wax and pollen, forming a cocoon in which pupation takes place. Filled circle, the life cycle of the queen is 15.5 days. Workers is 3 weeks and that of drones is 24 days. Filled circle, the young larvae are at first uniformly fed by a protein-rich diet secreted from the lateral pharyngeal glands of the workers. Then from 4th day onwards, larvae destined to become worker and male are fed with honey and pollen, and those destined to become queen are fed with the protein diet throughout. Filled circle, when the size of the population becomes excessive, swarms are emitted which consists of queen and workers. Thus the new colony is prepared for nest building and reproduction. Filled circle, after swarm, a new queen develops in the old colony. The new queen then performs the nuptial flight with the drones. After mating from mid-air, the new queen settles in the nest to start laying eggs. Filled circle, at the end of the summer, the workers ejects the drones from the hive as they have no further role in the colony. Developmental difference in early queen and worker larvae. Filled circle, duration of pupal stage, 7.5 days queen, 12 days worker. Filled circle, regression of ovaries. At 72 hours both castes have full set of 150 to 180 germinal primordia. At 96 hours, in worker larva, 4 to 5 primordia are left which develop into ova, whereas queen retains the full set. Filled circle, labial chemoreceptors. From 72 hours, queen larvae develop labial sensilia with 3 papillae while worker larvae have only 2. Filled circle fat body cells. During the first 3 days, worker larvae don't have fat cell reserves. Development begins only after 31 days when feeding of worker jelly begins. Queen larvae have glycogen and fat reserves from the beginning itself. Filled circle juvenile hormone. A high level of JH shifts the development to queen. This reduces the pupal stage. Regression of hypopharyngeal and wax glands take place. This stimulation of JH is mainly by the effect of royal jelly. Foraging behavior. Filled circle. A forager may prefer the nectar of one flower species producing greater quantities of nectar with a higher sugar concentration. A forager of a mellifera can carry as much as 7,080 milligrams of nectar per load. Filled circle nectaries can be located in various areas of the flower, and some species have extrafloral nectaries that may be visited by bees. In addition, some bees may perform nectar robbing, making a small hole at the base of a flower in order to obtain the nectar. Filled circle workers of all honeybee species carry nectar internally. Part of their alimentary canal is modified to form a honey sac or honey stomach. Filled circle. After returning to the hive, the forager regurgitates the nectar to one or more house bees, which then dehydrate the nectar and convert it into honey. Filled circle honeybee body are covered with abundant setae, which pollen grains are attached while she forage both for nectar and pollen. Filled circle. They make pollen to be pellets with nectar and carry them by pollen baskets on the hind tibia and store in the pollen storage area. Filled circle. In addition to collecting nectar and pollen, foragers can collect plant gum, propolis, and water. Propolis is a resinous hive product collected from various plant materials by honeybee workers. Filled circle. The flavonoids in propolis are considered to be responsible for its inhibitory effect on bacterial and fungus. Filled circle. Foragers communicate their floral findings in order to recruit other worker bees of the filled circle hive to forage in the same area. Filled circle honeybees communicate to each other by 2 ways. 1. Chemical communication. Pheromone. Filled circle pheromones are detected by the flagellum of the antennae. Filled circle. Most honeybee pheromones are produced by ectodermal glands of the epidermis and by mandibular glands. Filled circle. The mandibular glands are largest relative to body size in queens, large and well developed in workers, and very small in drones. Filled circle. The secretory product of workers' mandibular glands has an oily appearance, and its major component is 2-heptanone, a volatile substance. Filled circle. The pheromone either move through the air and are perceived by bee antennae or are perceived through direct contact with the antenna. 2. Physical communication. Waggle dance. Filled circle. Honeybees guide their nest mate for the distance and direction. Filled circle. The round dance is performed when the resource is within 50 meters from the hive. This dance consists of a scout bee, or returning forager, performing a series of narrow circular movements that may be repeated. Filled circle. Resources that are perceived as rewarding will have higher dance circuits performed. Filled circle. Similar behaviors occur for resources that are greater than 100 meters from the hive. These dances are more commonly known as the waggle dance, and encodes the direction and distance of the food source to her nestmates. Filled circle. Here the honeybee first walks straight ahead, vigorously shaking its abdomen and producing a buzzing sound with the beat of its wings. Filled circle. The distance and speed of this movement communicates the distance of the foraging site to the others. Filled circle. For communicating the direction, the dancing bee aligns her body in the direction of the food, relative to the sun. Filled circle. The entire dance pattern is a figure 8, with the bee repeating the straight portion of the movement each time it circles to the center again. Filled circle. The orientation of the dance correlates to the relative position of the sun to the food source, and the length of the waggle portion of the run is correlated to the distance from the hive. Also, the more vigorous the display, the better the food. Filled circle. The scout bees carry the unique smells of flowers they visit on their bodies, and that these odors must be present for the waggle dance to work. Honey production. Filled circle. Worker bees fly upwards of 6 miles per day and may visit as many as 50,000 flowers per day and gather the scant amount of nectar they need to make honey. Filled circle. Nectar is made of 3 sugars, sucrose, glucose, and fructose, plus some scent chemicals to help draw bees to the flowers. Filled circle. The nectar is sucked up using its long tongue called the proboscis. It is then swallowed into the bee's honey stomach. Filled circle. As nectar passes through the mouth, it is mixed with saliva and the enzyme invertase, which splits sucrose in the nectar into fructose and glucose, the sugars predominant in honey. Filled circle. Once their honey stomach is full, they fly back to the hive and transfer the nectar to other worker bees. Filled circle. The worker bees regurgitate the nectar, passing it mouth to mouth to the house bees. As the nectar is passed along, it is also chewed, which continues the enzyme mixing process. Filled circle. The hive's internal temperature is around 90.5 degrees Fahrenheit, which is warm enough that water in nectar begins to evaporate. Filled circle. The house bees begin to spread the mixture across the honeycomb. By increasing the surface area, moisture naturally evaporates, which reduces the moisture content. Filled circle. Bees fan their wings over the opening of the honeycomb cells. This helps increase airflow and speeds up the evaporation process. Filled circle. Once the water content is between 13 and 18%, down from 70% when it first arrived at the hive, the bees begin to cap the honeycomb. Once capped, it is preserved in the cells. Filled circle. Each bee will produce approximately 1/12 of a teaspoon within its lifespan, which is up to 6 weeks. An entire colony of bees can make around 5.4 gallons of honey per year. Filled circle. The kind of honey produced is dependent upon the plants they visit to collect nectar. Darker colored honey will often have a sweeter flavor profile than the lighter colored variations. Bee venom. Filled circle. Bee venom is mostly water -88%. Because of this water solubility, bees sting in moist tissue in order for the venom to disperse effectively. Filled circle. Bee venom is odorless with a pH of 4.5 to 5.5, which can make it slightly acidic. Filled circle. The allergen in bee venom is melittin. 50% of venom's dry weight comes from melittin. Filled circle. Melittin causes red blood cells at the sting site to burst and the blood vessels to expand. This blood vessel expansion is why some people have a dangerous drop in blood pressure after bee stings. Filled circle phospholipase A2 is another protein that works with melatonin to